Hello, my name is Justin Harris-Jennings, and it is great to be your instructor for your microbiology course. If you have any questions, feel free to email me. Before we get started, I wanted to let everyone know that I have functional movement disorder, which is a disorder where my brain kind of forgets how to function, and it causes physical and vocal tics. And so if something I say is kind of repetitive or there's some kind of tremor or shake to what I say, just know that that is a functional issue. There's no harm to me. It just time usually resolves it. If it happens, I'll usually address it and let you know that everything's okay and that it happened. But now that that's out of the way, I wanted to go through the syllabus real quick before we actually go into chapter 1 and 3 of this week's content. And so the syllabus is quite large. It's 22 pages. And I wanted to just kind of highlight a little bit of it. We can go over it in lab on Thursday. The first thing is that the Connect access is required and that you'll need a lab manual for the lab sessions. They will have some printed off the first couple of times. However, you'll need to get to the bookstore and acquire that lab manual. You'll also need a lab coat for PPE reasons. The first lab, you won't need it, so you'll have a little bit of time, but you can get a lab coat from Amazon. You could get an apron, for instance, even from Walmart, and that would suffice as just a PPE to provide protection from spills and things like that. There are colored pencils if you want your own, so you don't have to share. There is that might speed you up. And the Sharpie is helpful, but those things are not really actually needed. We'll talk about the attendance a little bit later when we talk about grades. The thing is with the exams is that they're proctored. And so, excuse me. And so you'll use Proctorio alongside of Moodle, and it will record your screen and record you as you go through the test. And it will like disable some functions of your computer. If you've already taken classes with this already, you might be familiar. There is a like setting up Proctorio or setting up proctoring in your Moodle section, and you'll need to go through those like steps before it actually lets you go and actually take the Unit 1 exam later this month. Then for your grades, this is the breakdown for the grades for the class, and these are the points that you're going to need, the point thresholds that you're going to need to get a minimum of those grade points. Just I know sometimes that's been helpful to have in the past. For the actual course itself, you're going to have 6 exams that are unit exams, and these are going to be delivered online and in person, where 70 points is going to come from the online and 30 points is going to come from the in-person that we take during lab session. We're going to then have a comprehensive final at the end of the year, and that's going to cover all of the 6 units that we learn about. And then you'll have throughout the year, each week, you're going to have a guided reading and homework section through your Connect SmartBook. And then at the end of the year, you'll have a lab practical where we go over the techniques that we've learned in lab. And throughout this semester, we'll have a couple of lab quizzes that will get points towards that. So altogether, it does I will want to say that in the past we've had complaints that these homeworks and readings take a really long time. I have gone ahead this semester and shortened those homeworks and readings, so they should be a little bit less burdensome on you as a student. However, with that, it's going to require you to go over less content. It's going to force you to go over less content. And so because of that, I'm hoping that's not going to create a deficit. If I do realize that that's hurting the class's performance, I would probably need to bulk those back up or figure out some other method to kind of bolster up that content for you guys. But that should be a little bit easier this year. but you do have them each week, and they are quite time consuming, and so they're best not to put off. It's also mindful of how many points they are worth each. They do seem like they're not very worth doing. That was when they were very long, so they should be worth more now. But they are worth a 1 unit, 1.5 unit exams together. And so individually, the assignments seem very small. However, when you look at them all together, they have quite a bit of your weighted grade. And so you'll need to be mindful of that. Throughout our lab sections themselves, you have these exercises that will complete, and those exercises need to be completed. If you don't complete them all, or you don't complete them within this threshold, it will start deducting from your lab quizzes total. So they only deduct points if you don't complete them all. So that's kind of a bummer that they're not like an add to your points, but they only take away if you don't do them. And similarly, your attendance also will hurt your grade. If you miss more than 2 lab sessions, you will actually have a reduction of 5% of your letter grade. And these are non-excused miss, just to be clear. If you have if you're going to miss or you are actively missing, just send me an email and let me know, and we can figure out if that's excused, if that's not, and how to make up the content that you missed. And so then the next thing is these numbers are going to be really important is as you get familiar with Moodle and Connect, these numbers are going to give us the best give you the best resource to resolving like any technical issues. There's also the Center for like Teaching Resources and Support and things, and that's going to be also helpful if you need anything. Quick questions, they're always available. Then I think it just says like what your expectations are. You can read through them. Expectations of use of AI is to use it responsibly and ethically. Try not to, like, you can read it actually, but like my biggest thing is use it ethically and morally and disclose your use. That's a big one. So then the next section, like I said, this is a long document. The next biggest thing is your course objectives. This is going to be like a really big place that you're going to come back to a lot, because these course objectives for Unit 1 are going to act as like the study guide for the unit exams. So I've gone through these unit objectives and I've specifically curated the exam to these objectives. So you can use these as a guide in your studying of what you need to focus on when you're going back over that content, preparing for your exams. And that there's the first unit is about how we kind of introduced microbiology, how we learned about bacteria and their structures. Then we're going to talk about in Unit 2, the biochemistry and metabolism, and then the molecular biology and genetics of bacteria. We're going to then talk about how bacteria can cause infection and disease in a host. Then we'll talk about the host response to that disease. And then we'll study specific bacterial diseases and then some viral diseases. And then in the lab section, we'll be talking about how to actually control these microorganisms and control them. If you scroll all the way down, you'll get to the schedules. So the first schedule you'll see is the homework assignment schedule. So the first homework assignment is this Wednesday, where you have chapter 1 and chapter 3 in the homework 1 and 3. And then if we go and you'll see the Unit 1 exam is going to be on January 30th. All of these are due at 11 p.m. And then if we scroll all the way down to the next schedule, we have the lab schedule. And so Tuesday, we don't have any lab meetings, but Thursday we'll talk about the introduction of safety in the lab and how we'll conduct ourselves in the lab. And then we'll go over the parts of a microscope and how to use them effectively. So with that, I think we can start the actual content. I guess I could go over the Moodle real quick. So when you get into your Moodle and you get into the course that you're wanting, you'll have the navigation bar on the left, and you'll have a start here. And that kind of has your syllabus. That's going to be where your syllabus is, and it has your schedules as well. I didn't talk about accessibility. If you have accommodations, talk to the accommodations office, the accessibility office, and we'll all work as a team to figure out how to best handle that. Then you'll have setting up online proctoring. In this section, this is where you're going to want to actually go through and set up and get a feel of how the proctoring is going to be set up, how it's going to feel to go through a test. So give me a second. There we go. So there are these instructions you can go through, and eventually you will come to the exam that you can take. Sometimes you'll get these messages here. Usually if that happens, you can click this secure exam proctor, and there's text that helps guide you through that. It'll say it's successfully installed, and then when you go back, usually this is resolved, or if you click into it, this will eventually say preview, and you can actually take that exam there. Outside of that, you'll have Unit 1. Unit 1 will then have your exams, your like recorded lectures and video content here. And then you'll have like Chapter 1 for your Connect, and you'll click that and it will take you to Connect, and you can actually go through and start that assignment there. So outside of that, we can start the actual content of Unit 1, where we'll go over Chapter 1 and 3, where Chapter 1 is kind of introducing bacteria, how we got to bacteria cause disease, but they also are all around us and help us in everyday life, and they can be advantageous to us in many different ways. How did we get to that? How do we classify them, and how do we write scientific names? And then in Chapter 3, we'll talk about different types of microscopy. And so in Chapter 1, we're specifically looking at microbes, and these very small, usually unicellular organisms that are typically prokaryotic, so they don't have membrane-bound organelles. And if you look at the evolution of life, these prokaryotes first showed up in the fossil record in microfossils and chemical trace fossils 3.5 billion years ago. And then eventually these diverged into more complex eukaryotic organisms, which have membrane-bound organelles, and then became multicellular, and eventually into reptiles and mammals that we see now in the past less than 1 billion years. So these prokaryotes have been around for an incredibly long period of time, and they've been able to colonize all reaches of the Earth in drastically different conditions, where you have some microbes that can survive drastically cold, frigid conditions, and some blastedly hot conditions with incredible pH levels, or even can tolerate salt levels. And so these microbes can come in different forms. You can have bacteria, and these are usually what you consider prokaryotes. You can have fungi, or you can have algae. You can have helminths, which are your worms. Your worms have a microscopic microbe stage of egg and larva, and then they develop into the big multicellular worms that you would find in your muscle, for instance. Gross. Or you could have viruses. Of note, viruses, because of quite a bit of different reasons that we'll cover in Unit 6, they're not considered alive. You'll typically talk to them talk about them as either active or non-active, actively replicating, non-actively replicating, or inactivated. And it's because they can't host their own biochemistry. And so typically viruses are considered complex viral particles, see, and they're simpler and they're have to depend on the host machinery and their like proteins and their biochemical reactions to replicate themselves, assemble themselves, and release themselves into the environment to then be spread to other things. And so it really, by themselves, the viruses can't do quite much. And so they're considered not alive by quite a bit of the scientific community. And so then we have excuse me. We then have these microbes that we've like actually used for a very long period of time. So they've been around, they can go into all of these different environments, but also humans have been able to utilize them for a very long period of time. So they've not only been in the environment, I'm just saying things in a circle here. These bacteria have not only been in the environment for billions of years, but they've also been alongside humans along that time. And so they have colonized us just the same as they've colonized the environment. And so they've colonized the inside of our gut, and we rely on them in many different ways to help digest food, metabolize those nutrients into smaller components during your like first like phase metabolism. You then have a lot of those bacteria that are actually making your hormones and vitamins that we're going to absorb up into to help elsewhere in our body. And so microbes in our gut have quite a bit of help, and we'll eventually talk about how even bacteria on our skin are incredibly helpful for deterring pathogenic, unhelpful bacteria to be on us. We also have been taking advantage of fermentation and the fermentation of microbes for tens of thousands of years. I think 12,000 years is what I looked up earlier. And so we have been able to make breads and like alcohols, and there's other things that are ferment that I can't remember. Breads, wines, and other things. And those things we have to utilize these microbes and take advantage of their biochemistry and their metabolism pathways to make ourselves specific food products or industrial products. With more modern technologies like biotechnology and genetic engineering, we're able to actually manipulate what bacteria is able to produce so that we don't have to so that it's not so like cost ineffective or something like that. So for instance, insulin is made from pig pancreases. That was its main source for many years. You would harvest many pig pancreases to generate this product of insulin. That's to give and treat diabetes. However, with genetic engineering, we've been able to make bacteria that is capable of generating this insulin in a satisfactory way and in like giant vats. So then it's now saving costs, farming those animals, harvesting those animals, and then there's also the ethical considerations of those animals not being involved as well. And so with you also have like cheese making. Oh, that was the other thing. Bread, wine, and cheese. Yeah, we've been doing that for a long time. We've been doing that for a long time. And so we've been able to like leverage these basic mechanisms that are found within bacteria to our advantage in a wide variety of our lives. We also can use them to help remediate environmental disasters like oil spills. Some bacteria can break down the polymers, the hydrocarbon polymers of oil. And so, or we can enhance that through genetic engineering, and then we can treat environmental spills and restore that to help clean that up. And I'm sure there's other toxic things that you can also deal with. There are specific like bacteria that like utilize iron in some way. So I'm sure that there would be like some capacity you could use that in like mining cleanup or something. Just kind of throwing things out there. But at the same time, there are a lot of different bacterias that have grown alongside of us and have not been helpful. They've kind of gone the other direction and have decided to be pathogenic. And so they've continued to grow alongside of us, but made us sick along the way. And they do this by not only maybe getting into our gut, but then maybe releasing toxins to then infiltrate into tissues that they're not supposed to get into. And so a lot of these diseases have been with us for just as long as we have been around, and the similar things with viruses, but right now we're talking about, well, I guess we're talking about bacteria and viruses both. And so, sometimes there are specific pathogens that are species specific because of how closely related these pathogen host interactions have fallen. But what's found is that over time with study, we found that a lot of these microbes cause diseases that we've seen clinically. And we see this with things like tuberculosis and ulcers and other things like I can't think of anything right now. Toxic shock syndrome, strep. Like there's literally so many. Most diseases. And so, but we've also been able to find that a lot of these chronic conditions that we see medically also seem to be associated with bacteria or viruses. And we see this most historically with gastric ulcers or stomach ulcers. These are like holes within the lining of your stomach. And so your stomach acid can get to the endothelium rather than be protected from the mucus. The mucus is kind of thin. And so the acid kind of damages the stomach lining. And so that hurts, it causes nausea, it causes pain, those kinds of symptoms. But traditionally, historically, it was thought that these ulcers were caused by stress, overworking, over exerting yourself. And eventually it came to a scientist or a physician's attention that these Helicobacter pylori bacteria, which are these spirocheque bacteria that are spiral shaped, seem to be in a lot of these patients that had these gastric ulcers. And so he took a vial of it and swallowed it and took it into himself and gave himself ulcers. And then he cured himself of those ulcers. And that's how he proved that these gastric ulcers were caused by this Helicobacter pylori. Thankfully, there are regulations and ethical guidelines that prevent this kind of experimentation. Currently, however, just don't. If you're ever in the situation. And so there's other ways to do it. But early in the time, we didn't know what was causing a lot of these diseases, and so along came this idea of the germ theory of disease, is that microorganisms can cause these diseases, because at the time they barely even realized that these microorganisms even existed. And so first you have Antine von Leeuwenhoek, who creates these really good lenses who can see these microorganisms and calls them animalcules, and or like animal molecules, essentially, really small animals. And he sees them, he's telling people about them, and other people are taking note of these same organisms. And his microscopes are some of the best, and he can get up to 300 times. Um, and a lot of these, oh, where were we? Germ theory of disease. We're still trying to figure out what's causing these disease clinically. A lot of the time we're thinking of these imbalance of humors or miasma or bad air that's causing these diseases that just are inherent to the environment, that are just or your actions. You've done this because of God's will and punishment. Those kinds of ideas that have led to, they were like, these things cause disease, but then at the same time they're saying, oh, these little microbes exist, these little animalcules exist, and eventually you're starting to realize that certain species of bacteria exist when we see some of these diseases. We're starting to make these tiny bits of connections, and around this time we're starting to develop the scientific method of how do we prove and actually solidify these connections of is this bacteria actually doing anything with disease? Is the bad air doing anything with this disease? Is it your imbalance? Is it because you got too much yellow bile in your stomach? Like, what's making you sick? And so a lot of these scientists and physicians are starting to go over and try to systematically figure out a way to prove these things. And they come up with the scientific method of making an observation, making a question based on that observation, which leads to a hypothesis that is a testable question. And we can make predictions from this hypothesis. If it's true, we can test it, right? And so we can then make predictions and test it and run an experiment to see if it is right. And this is the method that people started to use to determine that bacteria are causing disease and start to establish these foundations of the germ theory of disease. We have eventually the scientific community kind of comes up against this spontaneous generation of like, where are these microbes coming from? And again, are they causing disease? And so you have these things, these people like Francesco Redi, who determine that they find things that are like, oh, so I guess the first for aside, at the time, they didn't just think these things about microbes, but they also thought these things about bigger organisms, larger, complexer organisms. Like, for instance, we're going to talk about flies and maggots. They're going to talk about how they didn't know where these maggots were coming from. They thought they were just spontaneously generating on this meat. If you go back, like way back to like Greek philosophers, which is like further back than what we're talking about now, but like they talked about like eels coming from like mud, for instance. And so this idea of spontaneous generation has been like ingrained within us for a very long time. Excuse me. And so Reedy came up and was like, oh, these maggots are coming up on this meat, but also flies are on this meat. Could these maggots be coming from the flies? So this led to a hypothesis of maggots appear on this meat because flies lay their eggs. And this is a testable hypothesis because if we can prevent flies from laying their eggs, maggots shouldn't appear. And so they cover the meat and maggots don't appear because the flies can't get to them. And this leads to this experiment, right? And they can determine that, yes, these maggots must be coming from these flies that can't access the meat. So through many other experiments that are happening at the same time, like Louis Jablaut also had one that was talking about, can microbes grow in broth? So more of like a microbe question rather than like the insect, right? Excuse me. And so he would be able to boil hay in some water or soak hay in some water. And over time, it would get really cloudy because microbes would grow in it. And you could see these with like a microscope. And so he would realize that microbes grow in this infusion. And he's like, why do these microbes grow in infusion? Because, right, the scientific method is make an observation, ask a question, form a hypothesis. These microbes are coming from the environment where they're spontaneous generating. He thought they were coming from previous microbes. So essentially, a microbe gave rise to a generation and a new generation of microbes. So microbes come from generations prior. And so that was the hypothesis that Jabloch took. And he decided if he killed all of the microbes, there would be no parents to give off a new generation. And that is how he would be able to tell if these microbes, essentially, if microbes begot microbes, right? And he, in fact, realized that if you boil this hay infusion and you cover it from the environment, it will remain sterile. If you let it have free range of the environment, it will have growth. And so something that's interesting here is that we now know that there's something in the air that's causing these microbes to grow. And so Swan and Swaltz decided to do this new experiment where we had these infusions that were connected to an air outlet so that there was some air exchange. But this time they sterilized the air using a flame underneath. And this allowed them to sustain a sterile culture where the microbes were killed, or whatever was in the air was killed. Apparently, there was some disagreement about what was actually happening. They were thinking that the heat apparently made the air like hard for the spontaneous generation to happen. But Pasteur was like, no, that was just normal. You just were sterilizing it really good. So he used a different setup where he had this flask where this had this really long swan neck, and it made it where air exchange was incredibly difficult so that any dust or particulate matter didn't get into the infusion. And so it would stay sterile until that neck was broke or until those particulate matters were like blown into the neck of the flask. So in the medical community, we're trying to realize that I don't know. Whoa, that was rude. All right, so yeah, I think I'm gonna skip this slide. Yeah, essentially, the work from these people were showing, and like they were using these microscopes to find microbes on our teeth, in our body, on our surfaces, everywhere. And we were realizing how we can sterilize them to make it more of a clean environment, and how we can keep mixtures sterilized. And how could we apply this into the medical field? Different medical professionals started to discover, like how I said, I'm going to skip this and then completely went over it. So then there's Robert Koch, and Robert Koch, he developed these postulates of actually how do you link a specific bacteria to a specific disease. And so he creates a list of these. This is how you do it. Here's the procedure. And we'll talk about what that procedure is in a bit. And then Joseph Joseph Lister, kind of think of Listerine, he invented the aseptic technique of how we can actually clean our surfaces on our hands and things like that, our utensils and our surfaces, and how can we handle bacteria like cleanly without contaminating things. We then have Dr. Holmes and Semswell that learned about that they noticed that infants and mothers had high mortalities at childbirth, and that this was more often the case when they came from the autopsy room or the morgue. And so they had then set into place like hand washing procedures and found that that improved the health outcomes of these women and infants that were being birthed. And so we're starting to learn again, if we can clean our environment and control when and where bacteria are, we're able to do things more safely. Then Koch makes these postulates. He studied under Pasteur, and he links anthrax to anthracis, Bacillus anthracis, and he makes this procedure essentially is if this bacteria is infectious, and he does this in a couple of steps. The first, is this bacteria found in every case of these patients? Can we culture this in a laboratory? Can we then inoculate from that pure culture that we took? Can we inoculate a healthy individual and induce the same disease? Can we then reculture that bacteria from that individual? And so ethically, this isn't very easy to do because you can't just infect, excuse me, you can't just infect another person with a bacteria to see if they get the same disease. That would be incredibly unethical and dangerous, right? Otherwise, feasibility wise, not every pathogen is culturable in the lab. And so sometimes you just have to have genetic evidence that that bacteria is present, or you can use like a microscope and histology and immunofluorescence to identify them. So there are alternatives to culture postulates. There are more modern versions that are a little bit more feasible, but the same idea applies, right? You have to see that this bacteria is actually truly causing this infection, and it does so in every case, not just sometimes, because then it would just be like a contributing factor, for instance. And so it came to our knowledge that we need a classification system of how do we talk about and classify these pathogens and microbes. And so we have the Whitaker classification system, which uses your cell shape, the complexity of the cell type, if it's eukaryotic or prokaryotic, if it has no membrane-bound organelles, or if it has membrane-bound organelles, if it's unicellular or multicellular, and where does it get its nutrients type? Does it get it from its sun through photosynthesis, or does it get it from its other organisms through heterotrophy? We'll talk about, sorry, and for some reason I thought we were going over introduction again. And so Whitaker came up with this classification system where you have plants, fungi, and animals, which all came from protists, and those came from monorans. And monorans are your bacteria and your archaea. Your archaeas are these like more extreme versions of bacteria that's at like a very like naive level. However, this isn't the actual like agreed upon tree of life anymore. Just so we can see. The bacteria diverged earlier than archaea, and then you have those prokaryotes, and then you got your fungi, plants, and animals up here in your eukarya branch. And so this is what when you think of your evolutionary tree, this is what you want to view. But Whitaker at the time was talking historically, this was what they were going off of, where these bacteria and eukarya are these most primitive, and then you have prokaryotes that are more like multicellular, and then those diverged into our plants and fungus and animals. Those are those 5 kingdoms. And so we kind of go over this we use a taxonomy system to classify and name these organisms and how we can talk about how they are related to one another. When we use this level, and this taxonomy uses a hierarchy where things have a broad description and then a more exclusive description, where we have domain, kingdom, phylum, class, order, family, genus, species, where domain has the most variety. And as you get closer to species, it gets to one individual organism. Each individual organism has a 2-name nomenclature. This is a binomial nomenclature. So Homo sapiens would be the binomial nomenclature for humans. And these are unique to all organisms. They try not to be renamed. A lot of the time, these upper levels get renamed and shuffled as genetic information gets more fleshed out. And that evolutionary tree gets like reshuffled into a more like precise and accurate form as we get more information. One way to remember how this happens is, instead of just remembering domain, kingdom, phylum, class, order, family, genus, species, you can think of dumb kids play catch on the freeway, get smashed. That's the acronym that I always use since high school to remember that, and it's worked really well for me. There are definitely other ones. Feel free to come up with one that works well for you, but that one works well for me. So we then have the binomial nomenclature of how these organisms are actually written in things. We have this genus, and then we have this species, and typically we denote these very specially when we're writing, so that everyone knows that it's a species, that's an organism that we're talking about. And it does this by, when we're typing, using italics or underlining if we're writing. And after we use it a couple of times, we can shorten this to just a short E. coli. So this is Escherichia coli, and it is, or shortened to E. coli. And this is a coliform bacteria that's found in your gut or in the environment. And there's lots of different strains. Some of them cause disease and some of them don't. So getting into chapter 3, we can kind of talk about microscopy. And so we talked about Leeuwenhoek and how he did wonderful things for the microscopy world, where he had these lenses, and he had really powerful lenses and was able to see really small organisms with those microscopes. Today, we have microscopes that use more than one lens instead of just the one lens, and we actually have a lot of different parts in between that. So we have a light source that has this outsource of light that that gets focused down with the diaphragm. The diaphragm is just essentially a shutter that essentially makes it where we're not growing our light anymore. And instead, we just have this conical shape. The condenser focuses in this light down into a point onto our sample. And then our objective lenses, which are found here, which are changeable at different magnifications, then inverse the image, and they go through the ocular lens and then into our eye. And so this is how most microscopes work, and through different techniques and fanciness, you can get different aspects, and with a camera, you can get even better results. However, there is limits to light microscopy, like microscopy, and so you can only get so fine with your details until you can't determine, until things get too fuzzy. And so light microscopes have a resolution of 0.2 micrometers, and this is the distance in which two items are close enough that you can't tell that there is a distance between them. You can't tell if they're touching or if they're separate. And so light microscope has an actual threshold that is dependent on the wavelength of visible light that determines this like this optical limit or this resolution of the microscope. And there's different kinds of like microscopy. You have like compound microscopes, and you have standard light microscopes, and then we have bright field, dark field, phase contrast, and differential interference. And so bright field is what you'll see most of the time, where this is just shining a light straight through your sample. Your sample can still be alive in this system. And so you can just see right through it. You don't see much too much detail, but you can still see structures. You can then actually kind of put a little blocker in the middle of your light beam and create a black background, and this will let you kind of be more true to color, and you can kind of see more differences in structures. This again, you can have a live sample looking at this, and it can that would let you, for instance, like look at it and see it moving, for instance, or repeatedly look at it rather than it be like dead and fixed to a slide, like you would have like a tissue slide. Or in lab, we'll have heat fix slide where we've killed it with a flame. You have phase contrast microscopy, and this is going to look at specifically places where the density of materials are different. So if you think of when you mix water and oil together, they don't mix because they're nonpolar and polar. But if you look at their boundaries, you can see that there's a difference. They look different. One looks like oil and one looks like water, and you can see that they look different. And so those boundaries exist not only in materials that can't mix, but also they happen in materials that are of different density. You can think of like a really hot grill or fire where you have those heat waves coming off of it, and you can see how it's bending and how it's refracting the light differently. And so that is because the air is different densities. And so similarly, we can see that in cells where there are some portions of the cell that are more dense than others, and we can use different filters to like accentuate that. And so they do that with this phase contrast microscopy. So you can see kind of even greater detail of these structures and differences. Similarly, differential interference microscopy is a more complex technique that does similar things with these filters to create and identify these specific structures that are at different like densities within the cell. We then have fluorescent microscopy, and this is instead of using visible light, we can use lasers that are, instead of like white light, a very focused wavelength of light. One of the things that comes with this focused wavelength of light is we get actually improved resolution. So we can actually see things that are closer together than we would otherwise if we use just plain white light. And you can see this here where you can, where we have stained the entire bacteria here, or in this case, we have, I believe, individual bacteria within a cell with the nucleus stained. And so you can see quite a bit of detail of where of how these shapes are arranged, how they're oriented, where they're oriented, and what they're next to. And so fluorescent microscopy is incredibly powerful, and there's so many different ways to utilize it. It's a real neat thing. There's a lab that we get to do it, and it's that we get to use the fluorescent microscope, and it's really cool. We also have confocal microscopy, which uses essentially, instead of a 2D image like this, we get this kind of 3D stacked image so that we can see the structures in a more 3D volumetric version. You then have electron microscopy, and so instead of using a specific laser, we can try to throw smaller, essentially essentially wavelengths are just too big. And so the wavelength of the light wave is too big. And so electrons are a lot smaller. And so we can actually shoot beams of electrons onto a surface, measure how they bounce off of that surface, and recreate an image through electron microscopy and get 0.5 nanometers resolution, which is much higher magnification than we had previously. And there's two types of microscopy, electron microscopy. There's scanning electron microscopy, and this is kind of like scanning over the surface of something. And so you can see the surface of the cell, you can see how the surface of the bacteria and how they're being engulfed by this cell. While transmission microscopy is being shot right through the sample. The sample is real thin, and you can see in a 2D image the section of that sample. So with that, I believe that is all of the lectures. So I hope today that was helpful, and I am excited to look. I'm excited and for Thursday and look forward to meeting all of you. I hope this lecture was helpful. I will see you next time. Bye-bye.